Hello, Assalamu alaikum. Today, we want to continue talking about chapter 20, section 20.3, organization of electron transport complexes. Now, intact mitochondria isolated from cells can carry out all the reactions of the electron transport chain. 4 separate respiratory complexes can be isolated from the inner mitochondrial membrane. These complexes are multi-enzyme systems. Now, the electron transport reactions in the respiratory complexes occur. What reactions take place in the respiratory complexes? Now, you remember we encountered multi-enzyme complexes previously in chapter 19, namely the pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex. So each of the respiratory complexes can carry out the reaction of a portion of the electron transport chain. So what reactions take place in the respiratory complexes? We start with complex 1. This is the first complex, also called NADH CoQ oxidoreductase. It catalyzes the transfer of electrons from NADH to coenzyme Q or CoQ. It is an integral part of the inner mitochondrial membrane. It includes several proteins that contain an iron-sulfur cluster and the flavoprotein that oxidizes NADH. Now, the total number of subunits is more than 20. The flavoprotein has a flavin coenzyme called flavin mononucleotide or FMN. لاحظوا, it differs from FAD in not having an adenine nucleotide. And you can look at this figure, shows you the structure of FMN, the flavin mononucleotide. Now, the reaction of this complex occurs in several steps with successive oxidation and reduction of the flavoprotein and the iron-sulfur cluster. Now, step 1, transfer of electrons from NADH to the flavin portion of the flavoprotein. NADH plus H plus EFMN produces NAD plus E attached to FMNH2. Notice here that EFMN indicates that the flavin is covalently bonded to the enzyme, remember. So it is oxidized while it is bonded covalently to the enzyme. Now, in the second step, however, the reduced FMN is reoxidized, and the oxidized form of the iron-sulfur protein is reduced, as you can see here. Now, the reduced iron-sulfur protein then donates its electron to coenzyme Q, which becomes reduced CoQH2. So this is step 3. Then it takes place in 3 steps. Now, if we look at this figure, it shows us the oxidized and reduced forms of the coenzyme Q. Notice that coenzyme Q is also called ubiquinone. And this is the oxidized quinone form, and when it is reduced, it's called hydroquinone form. Then the oxidized quinone, the reduced hydroquinone form. Next, this is the overall reaction. This is the overall equation for the reaction. NADH plus H plus CoQ to give NAD plus CoQH2. Now, notice that The final electron acceptor in complex 1 is CoQ. This reaction is one of three responsible for proton pumping that creates the pH gradient. As I will show you in the next figure, the standard free energy change delta G naught prime is minus 19.4 kilocalorie per mole, indicates that the reaction is strongly exergonic and releases enough energy to drive the phosphorylation of ADP to ATP. Remember that we need only 7.3 kilocalorie per mole to phosphorylate ADP to ATP. And here we have more than enough. Okay. Now, remember that reduced NADH donates its electron to coenzyme Q, but not the other way around. Because of the so-called reduction potential, you remember NADH has a lower reduction potential than CoQ, so the transfer of electron from low to high. The final electron acceptor of complex 1, CoQ, is a mobile carrier. It is not bound to the, it is buried, it is found in the lipid bilayer. Let me show you this figure. This is the electron transport chain showing all the respiratory complexes. Notice here that the reduced cytochromes, the iron is in the iron 2 oxidation state. In the oxidized cytochromes, the iron is in the Fe+3 oxidation state. Now, we started here with complex 1. Okay, this is complex 1. Again, an ADH transfer its electrons to FMN, then FMN transfer its electron to iron-sulfur, which finally transfer its electron to coenzyme Q, as you can see. Now, NADH, remember that carry electrons and hydrogens in their reduced forms. But an iron-sulfur protein can carry only electrons. This is the basis of proton pumping that eventually leads to ATP production. And also this figure shows you the energetics of electron transport. Here we have 3 sites where proton pumping occur with ATP production. Notice that in each site, the transfer of electrons from NADH to CoQ produces enough energy. In cytochrome B, the same, and from A to oxygen also enough energy is produced to phosphorylate ATP to ADP, ADP to ATP. Okay. Now we shall see the second complex, which also transfer electrons from an oxidizable substrate to coenzyme Q. Now, so complex 2, the second of the 4 membrane-bound complexes called Succinate-coenzyme also catalyzes the transfer of electrons from succinate to CoQ. لاحظوا. Also here we have 2 stages, or 3 stages. Now, succinate is oxidized to fumarate by a flavin enzyme. لاحظوا. You remember succinate, the substrate from the citric acid cycle, is also oxidized to fumarate by a flavin enzyme. Now again, the notation EFAD indicates that the flavin portion is covalently bonded to the enzyme. Now the flavin group is reoxidized in the next stage, where as another sulfur protein is reduced, as you can see here. Then we have in stage 3, the reduced iron-sulfur protein donates its electron to oxidized CoQ, and CoQ is reduced to CoQH2. And the overall reaction is this: the oxidation of succinate to fumarate using coenzyme Q, which is oxidized to CoQH2. We already saw the first step of this reaction as we discussed the oxidation of succinate to fumarate as part of the citric acid cycle. You remember the enzyme traditionally called succinate dehydrogenase, which catalyzes this oxidation. Okay, of succinate to fumarate. Now, this enzyme has been shown by later work to be part of this enzyme complex, complex 2. Recall that the succinate dehydrogenase portion consists of a flavoprotein and an iron-sulfur protein. The other components of complex 2 are a B-type cytochrome and 2 iron-sulfur proteins. The whole complex, again, is an integral part of the inner mitochondrial membrane. Now, notice that this reaction is exergonic with small value for delta G naught pi. Energy from this reaction is not enough to drive ATP production. You remember I said we need 7.3 at least. So no proton is pumped out of the matrix during this step. Then, as I said, ATP production is coupled to proton pumping. So in complex 2, no proton pumping, no ATP production. Action of cytochromes. Now, in further steps of the electron transport chain, electrons are passed from CoQ, which is reoxidized, to cytochromes. Now, what are cytochromes? Groups of heme-containing proteins in the electron transport chain. In each heme group, the iron is successively reduced to iron 2 and reoxidized to iron 3. Also, the different types of cytochromes are distinguished by lowercase letters A, B, C, and further distinction is made possible with subscripts like C1. Then we go to complex 3. The third complex is called CoQH2 cytochrome C oxidoreductase, also called cytochrome reductase. So it has two names. Also called cytochrome reductase, catalyzes the oxidation of reduced coenzyme CoQH2. Now the electrons produced by this oxidation reaction are passed along the cytochrome C in a multi-step process. Now proton pass out on the other side of the membrane. And this is the overall reaction. Now 2 molecules of cytochrome C are required for every molecule of coenzyme Q. Why? Because the oxidation of CoQ involves 2 electrons, whereas the reduction of iron 3 to iron 2 requires only 1 electron. So that's why 2 molecules of cytochrome C are required. 2 molecules of cytochrome C are required for every molecule of coenzyme Q. Now, components include 2 B-type cytochromes, BH and BL, cytochrome C1, and several iron clusters protein. Again, if we go back and see that figure, this is, you see, complex 3. And this is complex 2, this is complex 3, and here complex 4, which we will see. Now this figure shows us the composition and locations of respiratory complexes in the inner mitochondrial membrane, showing the flow of electrons from NADH to oxygen. لاحظوا, complex 2 is not involved and is not shown, only complex 1 with 3 or 4, as you can see here. And this arrow shows us the direction of flow of electrons. Now this is CoQH, as I told you, is found in the lipid bilayer, it's mobile. And also cytochrome C is mobile, but it is located in the intermembrane space. We will see this again as we go. Now, series of reactions in the electron transport chain provides the link between two-electron transfer and one-electron transfer, involves the flow of electrons via a cyclic path from CoQH2 to other components of the complex, depends on the fact that coenzyme Q can exist in 3 forms. And these are the 3 forms. As I said, CoQ or ubiquinone, the oxidized or quinone form, then loss of electron, it becomes semiquinone, then another electron, it becomes reduced or hydroquinone form. And this figure shows us the oxidized and reduced forms of complex Q, showing the intermediate semiquinone anion form involved in the so-called the Q cycle. Don't worry about the details of the Q cycle that are found in the text. Now, in this part of the cycle, one electron is passed from CoQH2 to the iron-sulfur clusters to cytochrome C1, leaving coenzyme Q in the semiquinone form. And this is the arrangement, CoQH2 to iron-sulfur to cytochrome C1. And this is the equation. Now the semiquinone, along with the oxidized and reduced forms of coenzyme Q, participates in a cyclic process in which 2B cytochromes are reduced and oxidized in turn. Each of the 2 molecules of coenzyme Q involved in the cycle loses 1 electron. Again, complex 3, the Q cycle, provides a mechanism for electrons to be transferred one at a time from coenzyme Q to cytochrome C1. Now, complex 3 results in proton pumping and supplies enough energy. Remember, this is delta G naught minus 8.2 kilocalorie per mole to drive ATP production because of the reaction that it catalyzes. Recall that the phosphorylation of ADP requires 7.3 kilocalorie, and here we produced more than that. So again, in complex